office. And we work together at UCSF in the same lab, which he still uses, and he uses the lab as his Zoom background, as you can see. And oh yeah, for those of you who took molecular cell biology with me, and we did that family case study with the bioinformatics activity, I stole that from him. He was the one who actually came up with the activity. And I just took ownership over it. I'm kidding. I acknowledged you in the
Oh, okay.
handout. I put like from Vysotskiy
At all.
at all. Or was it just you? I don't remember. I think I just put your name there.
Yeah, I stole it from someone else also. That's how these
That's
things work.
how science works, right? So let me make sure I give you the sharing abilities. So Misha is doing his PhD at UCSF. I always forget the program that you're at. Is it Pharmacogenomics?
Yeah.
Okay.
Do I have it? If I Let me try to put up the slides. It might be on them.
Oh yeah, Pharmaceutical Sciences and Pharmacogenomics.
Yeah.
Okay, so I'll mute myself and I'll let you talk.
Okay. Cool. Yeah. Thanks for taking a break from refreshing election results to come to this presentation. Maybe this sort of is a topic that's a little bit different from what you're studying. I'm hoping that it'll still be accessible. Just so you know, I'm a bioinformatician. So I work with genetic data. I don't work with cells or anything like that. This presentation is going to be about data and how we can integrate it to solve biological problems. So the general biology that I'm going to be talking about is copy number variation, right? So in most cases, right, for every gene, we have two copies, one from mother and one from the father. And sometimes, like in all of biology, there are exceptions to these rules. So sometimes you will have a segment of your DNA deleted. So if you take this person on the left compared to this person in the middle, right? This person on the left only has one copy of the gene C, and this person on the right has a duplication, so they have a third copy of this gene. And when we look at brain-related traits, which is something that our lab focuses on in the Department of Psychiatry and Behavioral Sciences, we find that these kinds of mutations are very common in brain-related traits. So I'm going to be talking about two of them today. One of them is on chromosome 16. It encompasses about 30 genes. So here we either have all 30 genes that are missing or all 30 genes that are duplicated. So a deletion carrier has only one copy of each of these genes, and a duplication carrier has 3 copies. And a non-carrier, right, would have 2 copies of each of these genes. And so people who carry deletions in these 30 genes tend to have autism, obesity, intellectual disability. People who have duplications in these genes tend to have autism as well. They actually tend to be underweight, and they also tend to have intellectual disability, and they're also much more likely to have bipolar disorder and schizophrenia. Uh, the second region that I study is on chromosome 22. So 22q11.2, just to be clear, is a sort of a way to assign coordinates to to genomic regions. And so deletions in this large region, so people who are missing 60 genes, tend to have schizophrenia, autism, bipolar disorder, obesity, intellectual disability. Duplications also tend to have autism and intellectual disability, but people with duplications actually seem to have decreased risk of schizophrenia. So that's just kind of an interesting a thing that comes up here where sometimes deletions and duplications have the same traits, sometimes different, and sometimes it's just the opposite. Now this is a very well classified mutation. It's also called DiGeorge syndrome and velocardiofacial syndrome. Maybe that's a term you've heard somewhere. And has a lot of non-behavioral features, like, for example, heart conditions. So a lot of people who have deletions here have various malformations in the heart, as well as in the face. But for the most part, I'm going to ignore those and focus just on the behavioral brain-related traits. And so this is the overall question that we're going to try to address, is that we have this region with a lot of genes, and probably not every single one of these genes contributes to every single one of these traits. So what we want to do is to basically um, figure out a way to assign each of these genes. So assign which is a gene that leads to autism, which gene to bipolar disorder, which genes might drive both, and which genes might drive neither. Um, so why is this kind of a difficult problem? Well, one reason is that there's no clear critical region, what we call it. So sometimes what might happen is that we'd have like 10 genes that are deleted, and what it and in some patients we might find, well, actually, there's two patients with two genes within that deleted that have exactly the same phenotype. So in those cases, we are able to say, okay, those two genes are important, because they seem, sufficient to cause these traits. For these regions that I'm looking at, that is not something that has been found. Same with disruptive mutations. Again, if there is kind of a mutation in one of these genes that was consistently causing the same trait, then we know that's the gene to look at, but that has not been found for my genes. People have studied this in animal models, in mice, Drosophila, zebrafish. And what unfortunately happens is that different animal models and even different experiments with the same animal models are coming to different answers. Um, and interestingly, mouse models seem actually flipped from human behavior. So for example, at the chromosome 16 region, 16p, what we see in humans is that people with deletions of that region tend to be more likely to have obesity. And the opposite is true in mice. In mice, deletions of that region are more likely to be underweight. So that makes it kind of difficult to use mice as a way uh, to analyze these sorts of human traits. And at the same time, some of these brain-related traits, are difficult to measure. So, for example, right, if we want to diagnose schizophrenia in a human, right, one of the hallmarks would be hallucinations. But in mice, that's just not a trait that we would be able to measure. So here comes the sort of the idea that's driving this project, and that has to do with transcription and RNA, which hopefully you've talked about either in this class or another. And so to take a look at this chart, at the top, where we have deletions, people with copy normal and duplications. So if you have people who are copy normal, meaning they have two copies of each gene, they're going to have some sort of intermediate amount of RNA that's going to be transcribed from these genes. People who have deletions and are missing a copy of a gene, they're probably going to have less RNA that is coming from that gene. And people who have duplications, since they have an extra copy of the gene, they're probably going to have additional RNA transcribed. And of course, the more or less RNA, the more or less protein you have. Um, so so that's the sort of the idea that we're working in. Now, we know that this is actually happening because people have measured RNA levels of people who have deletions and duplications in humans and mice. So as you can see, this is a chart on the bottom. You can see that in the middle is the CNV, the copy number variant. Uh, so the middle here are the genes, and above the line are duplications. So people who have duplications of this gene have increased amount of expression. People who have deletions have a decreased amount of expression. So we have this sort of evidence that this kind of model might hold true. Now, the other idea that I want to bring in here is that gene expression levels vary regardless of copy number. So for any individual gene, right, all of us here are going to have a different expression level of that gene just naturally. And some of us, go on to develop schizophrenia and some of us don't. Um, so we can consider this sort of model. So as you can see, say in the middle is the distribution of a gene expression for a non-carrier. On the right is for a duplication carrier. On the left is for a deletion carrier. And so duplication carriers will on average have higher expression. Deletion carriers will on average have lower expression. And non-carriers will have some other range of expression that'll intersect duplication deletion carriers, but will probably not be as extreme. So consider person B. So person B does not have deletions or duplications. Person B has some very average gene expression of that gene. Person C, on the other hand, has increased expression of this gene. Person C, again, does not have a deletion or duplication, but person C's expression looks more like a duplication carrier than person B. So if this model holds true, then person C should have a higher risk of developing schizophrenia than person B, because the expression of its gene is higher. I'll get into now kind of the more technical methodology that we address this question with. And the idea behind this methodology is that for a lot of these traits, we have people, tens of thousands of people who have genetic data that are measured, that have been genotyped. But we don't have that many people who have transcription level data, especially in the brain for these genes. So the question that I'm going to, or the question that this methodology answers is, You know, how do we use this genetic information to get a gene expression? And this is a methodology called predict scan. And the way it works is like this. We take these control group of people, just healthy people, and we have their DNA and we have their RNA levels. And what can be done with machine learning is to make these sorts of predictive models where, given a certain genotype, we can predict the RNA expression level for that gene. And to do this, we use the genotype tissue expression cohort, or GTEx. I know maybe Eric has mentioned that. I'm not sure. Where they basically, it's a consortium where they measure genetic and gene and RNA information for like 50 different tissues in control individuals. So these predictive models are done, across various tissues, including brain. As you can see by number of samples over here are the brain samples, and their sample sizes are a little bit smaller than some of the others. So in practice, we actually use all tissues together. But that's kind of a technical detail that I won't go into. So then, we can take genotypes from a disease or, any trait of interest, and then apply these predictive models. And then you get out the predicted expression of this gene. And then finally, you'll end with a statistical association test, kind of like GWAS. I know if you've covered that or not, but basically, we take predicted expressions and phenotypes, so has schizophrenia, non-schizophrenia, and we test for each gene, do expression differences in that gene have an association with schizophrenia, or again, any trait of interest. Uh, Again, this is a technical detail, but if you have GWAS results, so genetic association results, you can skip some of this part. Um, so here are some of the large genetic data sets that we have. These are from the Psychiatric Genomics Consortium, which is kind of a worldwide group of labs that study these traits and pull together various data sets. So these are the sample sizes. Schizophrenia has largest sample size. It's actually still smaller than what would be great, but it's a fairly large sample size. Autism is still nearly 10,000 of controls and 10,000 cases, but slightly less than what would be perfect. And these are separated into cohorts. The methodology. I'll go through quickly. You can ask me more about it later. But we do this analysis combining across tissues, and we also get the average direction across tissues, because we're kind of interested in direction, and we do some multiple testing correction. So this is what the results look like. So this is sort of like a two-sided Manhattan plot, if you've looked at those before. But on the x-axis, we have the list of genes. So this is for the 22q region on chromosome 22. We have a list of genes at the bottom, just kind of in order of where they appear on the chromosome. And then on this axis, you have a z-score. So that's a metric of statistical association. Um, and these dashed lines represent the significance threshold. So if a z-score is surpassing one of these lines, surpassing significance threshold, then this gene is significantly associated with this trait. Long story short, no genes at the 22q region were associated with any of these traits using our methodology. However, what we did find was in schizophrenia on chromosome 16, there were actually a few different hits. And I apologize, actually, the slide is actually a little bit outdated, but in general, we have several hits, with the top one being INO-ADE, and then a few kind of other genes. If you remember what I said at the very beginning, schizophrenia is associated with duplications of these regions. And this is what we see here, actually. For these genes, we see a positive association. The numbers on the y-axis represent direction of association, a positive association between the expression of these genes and schizophrenia. One question that we might try to get at is how many of these signals are unique. And again, this might be a little bit of a technical detail that's beyond the point, but what happens is that when we do the prediction model training step, the SNPs that go into these predictive models tend to have various correlation patterns. And so when the SNPs that go onto these models are correlated, well, these predicted expressions are also going to be correlated. And so some of these signals might not be independent. So we took an approach to figure out how many of these genes are independent. And basically what we did was, for each gene, we adjusted the predicted expression of this gene by all of these other 4 associated genes, and vice versa, to see, what happened. This is what we call a conditional analysis. So to see what will happen if we take a gene of interest and adjust it for the others. And so this is what we find. So we take, what we found was that we have this one gene, INO-ADE, that when we condition on the predictive expression of the gene, suddenly there's no other genes that are surpassing this threshold. But if we keep in INO-ADE and condition out all of the other genes, well, we see that INO-ADE is still significantly associated with schizophrenia. So that what that implies to us is that INO-ADE is the sole gene in that region that drives schizophrenia. We also did a replication analysis. So we took an independent data set from the UK Biobank. So that is basically a resource of several hundred thousands of people in the UK who have been genotyped and phenotyped. So it's not actually a huge amount of people in this biobank who have schizophrenia, about 200, which I think is still about the population prevalence. So schizophrenia is about 1% of the population. So, 200 right out of, whatever, like 200,000. I think that works out. And we see that even in this additional data set, INO-AD does replicate at a p-value of 0.04. We also looked at 2 more traits, IQ and BMI. So IQ is a proxy for intellectual disability, which is a trait associated with these deletions, duplications. And what we found is that there's one gene, SPN, on chromosome 16, that is very strongly associated with IQ. And this is in a very large data set of over 200,000 individuals. IQ is associated with both deletions and duplications of this region. So, we don't really care about direction. We also looked at BMI, which is a proxy for obesity. And we found that there were 4 genes that were associated in the negative direction. So obesity is associated with deletions on chromosome 16. So that's why I was looking specifically at the negative direction here. And so we found 4 genes that are associated with BMI in the negative direction, SBN, TMEM219, TAUK2, and INO80E. An additional 2 other genes that were associated in the positive direction that we did not expect. Those were MVP and KCD13. What I'm not showing here is that I did the same sort of independence analysis, and I found that of these 6 genes, 3 of them were independent: INO80E, SPN, and KCD13. So what we see actually is that, we know that at least, multiple genes can drive the same traits. So we have now INO80E as a driver of schizophrenia and BMI, and SPN that's a driver of both IQ and BMI. So this is kind of the first part of this approach. For the second part of this approach, we integrated electronic health records. So this is, I think, kind of a big part of this study is, as I said, entitled is integration of genetic and clinical data. So at Vanderbilt University, there is a hospital, and attached to this hospital is a biobank. There they have a sort of opt-out system that every patient in the hospital, is intended to get their genetic sequence. So right now there's 3 million patients, and there's about 48,000 people who so far who have gotten their genetic sequences. These biobanks are annotated with these values called Phi codes, which just represent clinical traits. So Phi codes are very often similar to things like ICD-10 codes and ICD-9 codes that you might have heard about that are used in medicine. They have their own sort of version in their biobank called BioView. And so one of the first things that we actually looked at was when we looked at this biobank is that actually some people in this biobank had 16p11.2 mutations, duplications, deletions within their medical records. So what we did, and this was done by Tyne at Vanderbilt, was that she queried all of the medical records for various terms. So 16p11.2 duplication, 16p11.2, gain 22q11. And she literally went through and searched through all of the matched records to confirm, how many people's, mutations seem to be relevant to us. So we excluded certain, people who had 16p11.2 in their medical notes, but we couldn't really tell if it was duplication or deletion. We excluded people who had multiple genetic mutations. And what we found was we had about 50 within the biobank people with 16p duplication, deletion, 22q duplication, about 400 with 22q deletions, which makes sense actually. The 22q deletion is a fairly common mutation, appears in about 1 in 3,000 individuals. And a lot of people that are born with, heart defects might get tested for this mutation. So that's why we have more of these. We also separated out people who were and weren't genotyped. So within duplication, 16 duplication deletion groups, there was 48 people that had this duplication deletions but did not have genotypes. Um, and so we want to separate out the genotypic and phenotypic analysis. And the question that we asked was, so for people who had the CNV mutation, what are the other traits that these people are more likely to have? So this is an approach called PheWAS, the Pheno-Wide Association Study. And so what we do is we take these people who have the CNVs, and then we have controls. So they used about 700,000 controls. And then we go through every phenotype that's listed in the biobank, which is about 1,800. And we compare, for all of these 1,800 diagnoses, which are the ones that seem to be overrepresented in cases versus controls. You'll have to, bear with me since for some of these slides, um, uh, but this is what we get for 16p duplications and deletions. And just to kind of briefly note, so on the x-axis is different phenotypes. These labels you see are categories of phenotypes, so like infectious disease, congenital anomalies, sense organs, and everything above this top line are phenotypes that are significantly associated with these duplications and deletions. And what we find is phenotypes are both known and novel. So for example, here at 16p deletions, we have autism, mental retardation, um, you know, uh, muscle weakness. Those are all known phenotypes of 16p deletions. We also find things like sleep apnea, which is not a phenotype that's been observed in 16p deletions. Um, in duplications, we see similar phenotypes. We have convulsions, so epilepsy-related phenotypes that we were aware of, delayed milestones, and bacterial infection are not always specified. There's not been any evidence that people with these mutations are more prone to infectious diseases. So that's kind of a phenotype that we found that we did not expect to find. Same with so looking the same with 22q deletions and duplications. Um, so 22q deletions, we have things like cleft palate, cardiac anomalies, a lot of heart-related stuff, immune deficiency, cardiomegaly, a lot of these traits that, if you're a clinician, you would recognize, oh yeah, these are traits that we would expect to find with 22q deletion carriers. But also it's kind of like disorders of parathyroid gland, hypoparathyroidism that have not been described before. 22q duplications, not a huge amount of traits. We have hearing-related traits which have been known, developmental delays, delayed milestones. So in general, when we look at this biobank data, we can pick up the traits that we would expect to find on people who carry these mutations, and people who and traits that are novel. There's also a couple intriguing findings here. So in 22q deletion carriers, one of the traits that was overrepresented was dementia, 16p anterior hornstead disease. So this is kind of like ALS, and then cerebral degenerations in 16p duplication carriers. The average age of carriers in the biobank is under 18. well under 18 actually. And these are traits that we also found to be overrepresented that happen quite a bit later in life. So we don't know actually, is a 22q deletion a risk factor for dementia? Um, we just don't know that because the majority of the people that we study are just not at that age. Um, and so what this means is that we really need to have you know to have longitudinal studies to follow these people around and to see you know th- this person seems like they might be at risk of dementia or ALS, and we should be keeping an eye out for that earlier. And so the third part of the study is where we are going to integrate the genetic information and the biobank information. So this is again an approach called FEWAS, and so the approach that I first told you about was when we started with a phenotype, like autism, and we got all associated genes that are associated with that phenotype. Here, we're going to start with the gene, and we're going to get all associated phenotypes among 48,000 individuals using that same method of predicting the expression and see for which traits, you know, does, so here on this diagram, we have on the left people with high predicted expression of a gene, people on the right with low predicted expression, and we're going to compare the people with high predicted expression of a gene versus low predicted expression of a gene, which diagnoses are they most likely to have. And what we find are 17 significant gene trait pairs. So we find NPIB11 and SLX1B at 16p11.2 is associated with psychosis, and SCARF2 at 22q11.2 associated with mood disorders. Now, if you notice these are not the same traits that we tested earlier, right? We tested schizophrenia. We did not test psychosis. We tested bipolar disorder. We did not test mood disorders because in these large data sets, there we look at a very specific definition of schizophrenia and bipolar disorder. Here, we look at some of these more subphenotypes like psychosis and mood disorders, and that allows us to pick up genes that we did not find just looking at schizophrenia and bipolar disorder. And if you remember, at 22q, I didn't find any hits at all when looking at any of the large data sets. And here we found 3 novel candidates for these 2 traits that have not been found by anyone else studying these traits before. Um, Furthermore, and I won't go through this whole table, is so we can divide our traits into clinical categories. So there's like 17 different clinical categories, and take a look among the top phenotypes for each of our genes, how many fall into which of these categories. We can also, calculate an expected number of phenotypes that we should be getting, knowing how many of each category we're testing, and compare observed and expected. And so when you do a statistical test to compare observed and expected, we find that mental disorders are the only category that are significantly overrepresented in the FEWAS results of our genes, 98 observed versus 64 expected. There's no other categories that are overrepresented. There's one dermatologic trait that it's underrepresented among our genes. That kind of gives us a sense to see, yeah, we want to find brain related traits, and yeah, among all these traits that are being driven by our genes, and again, these are traits that are kind of on one hand specific, on one hand kind of broader, like psychosis, that those are the traits we're picking up the most. And finally, what I want to show you with these are circular plots, and you can look through them on your own time. This is a study that's currently on bioRxiv, where what I tried to do was basically to catalog the effect of each of these genes on what it might do. And so with the so at the bottom of these circles are the genes. On the top are phenotypes. And so the phenotypes that are included here are included either because they are brain-related traits, or because they're the same phenotypes that came up when we compared traits that are actually overrepresented in CNV carriers. So for example, on the left, and I know this font is, a little bit small, we have the first trait on the left is dysphagia, which was overrepresented both deletions and duplications. And we found traits that are associated with these. Infantile cerebral palsy was associated with duplications, and psychosis, a brain-related trait. Ultimately, the goal of when I was integrating these various studies is to take these genes that are either all duplicate or deleted and kind of catalog their their effects on various traits. To sum up, this is the integration, how we integrated the various data sets. So again, we were motivated by these copy number variant regions that are deleted or duplicated in people, and we want to study the effect of each individual gene. So the first way we did this was we took these brain-related traits, we took these large data sets and tried to find pairs of genes that are associated with these 5 traits: autism, schizophrenia, bipolar, IQ, and BMI. So at 16p, we found results for schizophrenia, BMI, and Q, not for autism or bipolar disorder, and none for 22q. Second, we went into this biobank and looked at medical records, and we compared different phenotypes there, and we recapitulated known carrier traits and found a few novel ones. And then we integrated both the biobank and the predicted expression by taking basically, the 1,500 phenotypes and performing association studies between the various phenotypes and the genes in the CNV to find overlap between PWAS and carrier traits, as well as brain-related trait signals. Actually, not weak ones, I think. When I first made the slide, they were weak. No, actually, now we have some very strong brain-related trait signals. The future direction now is thinking about the idea that, well, maybe one individual gene is insufficient to cause association with phenotype. And maybe it takes genes working together to drive the various traits. So right now, what I'm basically doing is making these combined genes. If you take gene A and gene B, that have certain gene expression. So these are numbers, are just, pretend that this is the RNA level gene expression of these genes. Make a combined gene where the expressions are the sums of these individual genes. And why that might help? Well, if you consider, between cases and controls, the difference here is, between controls and cases is what, like 3. But if we combine together two genes, 3, 4, this difference is now 7. So, now these differences between cases and controls could be much larger and allow us to pick up maybe signal that we just would not have been able to find looking at individual genes alone. I think that's it. Um, and so finally, as Erica asked, to talk a little bit about career. I don't actually have any sort of interesting career story. So my bachelor's was in mathematical and computational biology. And while I was doing the bachelor's, I did a summer internship at UCSF for 2 summers, and then I wrote my undergraduate thesis with Graham Johnson. The lab was called Mesoscope, and the lab was about basically modeling biological systems. Right now, Graham is not at UCSF anymore. I think he is at Brain Institute in Seattle. And the goal was to work on simulations of molecules. So the molecule that I was focusing on is kinesin, which is a motor protein. Maybe you've heard of those. And it's just kind of a protein that, just walks around on these two legs, basically taking uh, items from one side of the cell to another. And my goal was to figure out, a way to model the activity of this protein on the computer. And so the way we did this was through basically implementing this flowchart. So we basically, took the how this motor protein works and divided into kind of various steps. So for example, sometimes just free walking in the cytoplasm. Sometimes one motor, binds weakly. And once that motor binds ATP, it binds very strongly. And then, it basically does its little walk and switches feet. And so what I was working on is basically, yeah, how to model computationally this effect. I briefly also worked in a squirrel lab, but that's a story for another day. And that's it. And then I came to do PhD at UCSF, and I've been here for a very long time. I guess the final note that I'll give will be sort of about bioinformatics, that they for people who are interested in combining biology and informatics, in a broad term. um, you know just kind of talk about what sorts of career options there are. So there is academic, affiliated with a university. So that's, you're a professor, you're a staff scientist, you're a researcher, you're a PhD student, a postdoc. So those are one way that bioinformaticians can be academic. Academic, large universities also have these things called core facilities. So core facilities are where you are not actually doing any data analysis yourself, or rather, you're not solving any biological process yourself, but people from any lab will just bring you data that do not have the capabilities or capacity to analyze it yourself or themselves, and you will look at their data and provide output. There's industry, what we call it, right? So that's where you're affiliated with a company, and so there's a lot of different companies out there. Consumer genetics, pharma, basically people that make biological equipment. A huge list here in the Bay Area. And of course, entrepreneurs, you can have a great idea and you can start a company. So here's just kind of a list of problems that bioinformaticians solve, and bioinformaticians at various in various capacities, and I won't go into detail into any of these. If you want to ask me kind of afterwards to go into more details about any of these, I can, but this is kind of my field is this last one, which is genetics and omics. But there's plenty of other fields that bioinformaticians work in. And if you want to kind of start brushing up on your skills, you'd probably want to learn a programming language like R, Python. You'd want to learn a language like Bash or Unix. You'd want to learn some statistics, statistical linear models, especially, and then machine learning is everywhere. So that's something that you can look at also. And the question that I want to end with, is do you need a PhD? In terms of bioinformatics, I'll say specifically, if you want to be a professor and run your own research lab, you will need a PhD. In a lot of big companies now, you're not really competitive unless you have a PhD. So, if you want to work in pharma or biotech, PhD is it'll be highly desirable. Um, and one thing that I was kind of also thinking about recently, I forget if it's any of my remaining slides. Um, Sometimes a PhD is helpful even if you don't want to do science, right? Like there's plenty of career paths after PhD. So, for example, something like scientific communication. There's people, who um, do, marketing, for example, at biotech companies who really, really like PhDs, because there's always people who need to make a bridge between the scientific community and the public community. Even for some of these big companies, actually, even if you don't become a scientist, actually sometimes having a PhD can be helpful with that. But that's a little bit of an aside. Um, for a small, medium-sized company, that's kind of rapidly hiring, they might not be looking that much for a PhD. Um, for some of these core facilities in academia, when you're just sort of like, looking at data and trying to analyze data, you might not need one. If you want to start your own company, again, if you have a great idea, then you don't need a PhD, but you'll probably want to hire some. And you probably don't need a PhD if what you really like is the programming aspect. So, if you want to write code, if you want to write, software, for both academic and industry labs, you don't need a PhD for that. Or, just being a data analyst. So to go into data science, you do not need a PhD, although it can sometimes be helpful. That is all I wanted to say. So I know I'll open it up to discussion. If anyone wants to ask or contribute a point.
Well, thank you. Does anyone have questions for Misha? You're muted, Sayaka.
Sorry. Thank you very much for the talk. I'm sure everyone's PhD story is different, but how are you feeling about yours? Are you happy that you got it? Was it really difficult, or was it like just something you enjoyed throughout the whole thing?
I'm still in process. So
Oh,
so
sorry.
so yeah, I mean it's a lot of ups and downs, and I think and yeah, it is different. I like UCSF a lot. I think it's a great place to do a PhD. And right now I'm kind of yeah, I mean, I'm a scientist by nature, right? Like, I think that's kind of one of the first places where, you could be like, oh, I just discovered something that no one else has ever, learned before. And that's, very cool. Um, I am right now, I'm kind of looking at moving into probably something non-academic, so an industry position. And one of the reasons behind that is what I find, and this is my particular story, is the sort of like, and as a PhD student, it's a lot of, kind of on my own, is that, I run this project, I run it basically from start to finish. Um, and it's just me. And, so what I love to have more is, is this sort of like more team group environment. So that's kind of one thing I miss. But my lab is also tiny. My lab is a lab of 5 people. And so people, and you can decide about, what sorts of sizes, are appropriate for you. I like it small, but if it's small, right, there's these same drawbacks, but people, if you have like, 20 people, then, you might be more collaborative, but there's also plenty of other drawbacks for people who work in huge labs.
I'm just gonna bombard you with questions, but maybe you've mentioned this, but you were originally math and computer science major, but you went to bio. Is that like quite common, or with a leap of, oh, actually, like
So I didn't catch the last
interested
part of
in
that.
biology more than I thought, kind of thing.
So actually, my bachelor's degree was
Did I
just
freeze?
math, computer science, and biology. So I did study biology, and I knew I wanted. I actually knew since high school that that's the field I wanted to go into, integrating math, computer science, and biology. Um, how many people switch from purely math and computer science to biology? I think a decent amount. Because I think a lot of people, kind of want something to be more direct and relevant, right? So, you can program, for Apple, however much you want, but sometimes, people realize that they want to actually study something about humans and human health. Um, and yeah, I think it's not uncommon for people to switch fields into PhD. Usually not by a whole lot, at least not these days. It used to be very popular that people would start as in physics and then switch everywhere. But now, my background was more in biophysics and evolution, and for the PhD, I switched to more genetics. And there is a learning curve anytime you switch, but y- um, you know it's uh, you know part of PhD and even part of bachelor's, in my opinion, is teaching yourself how to learn and how to immerse yourself into a new field. And they don't teach you how to be patient with yourself when you're doing that. That's a skill that that is very hard to develop, but is also important.
Okay, one last question, I promise. I'm interested in animal welfare and animal behavior. And you mentioned that it's really difficult to measure like schizophrenia phenotype in mice. Do you guys actually try to measure that? Like, how do you even?
Yes, so in my lab we don't do that, but people who, but there's definitely plenty of behavioral people who work in mice. So something like depression, maybe you can understand a little bit easier. So for example, if a mouse, you know, suddenly doesn't want to eat, you know, if a mouse like, you know, if most mice are active and a mouse that's like very passive and doesn't care about eating or doing things, you know, we can kind of measure that as depression. You know, if we have a mouse that, you know, doesn't form social interactions with other mice very well, you know, that's kind of a sign of autism. You know, if we have a mouse that, you know, always stays in its safe space and doesn't go out exploring, you know, its cage, you know, that's kind of a sign of anxiety. You know, these are all kind of, you know, very proxy measurements that, you know, are will never recapitulate the full extent of these traits in humans. But you know, but there's definitely, yeah, people who try, and in, you know, in mammals, of course, it's a lot easier than in even other model organisms. You know, for things like zebrafish, you know, it's sort of size of brain and maybe how fast they're swimming is all you can measure. In mammals, it's a little bit easier.
Thank you very much.
What's your favorite animal model?
What's my favorite animal model? Humans. uh, no, I know. Honestly, I know this is a cop out, but computers, like, you know, that, you know, and computers, you know, are really hard to model things on in even different ways. Um, but in a, modeling things on the computer is just way faster than everything else. That you know, I could know within a day whether an experiment worked or not. Whereas in most other models, it will be, you know, days, weeks, months, more than months if you're really unlucky.
So you mentioned that your PhD life was up and downs. Do you mind sharing the uppest up and the downest down?
Um, um, let's see. I let me try to recall. I know. I I think um I know the upper stops I think are um actually situations like this where, you know, I get to you know, present my work to other people, other scientists, other non-scientists, um, and you know, have them you know, think it's cool. Um, you know, I know I'm a very I'm a very introverted person, but what I don't have is a fear of public speaking, and so all those kinds of situations are um, you know, the best. You know, the lowest is kind of, you know, when you're stuck. And, you know, as I said, it's kind of my project. It's me and my advisor who knows what's going on. And, if you can't figure it out, you're kind of on your own. So there's sometimes even weeks where you're just staring at the screen and don't know what to do and just waiting for inspiration. I think those are, yeah, big
Where does
ones.
that inspiration come from, if it ever comes?
Um, I honestly usually just figuring out an alternative way to do it. Scrap what I can't figure out, and just move on. Um, same with the writing papers. My boss and I, we have sort of different, ways we like to write papers, and different philosophies. So sometimes I'll write something and she won't like it, and I'll try it again and she still won't like it, and at some point I'll just delete that whole section, and then suddenly she likes it.
I know that feeling.
Yeah. I mean, I think it's universal that
Yeah.
not just that, but that, like, you know, you do something, the boss tells you to do something and you do it, and then they come back and say, oh, that's a horrible idea. And you're like, but you're the one that came up with that idea. That's that's not just true in science. It's actually very true in academia in general. But I think that's in all fields, that is something that you deal with.
You send out a paper, you're like, this is so good. And then it comes like all reds, all corrections.
Yeah. Uh, but I think there's a lot of perfectionists that end up in PhDs and in academic scientific careers, and we all have our own way of being perfectionists. And it's understandable. The further up you go, the more competitive it
Mhm.
becomes. Especially if you're working on really, really cutting edge things, it becomes very competitive. And when you're a professor, trying to get grant funding, it's very, very competitive. So that's kind of the philosophy of that. My goal was never to be like a fully research professor. My goals have been either to work in a company, um, or to being a professor in a school that also has a teaching focus. Um, and if you're if you're interested in having any sort of teaching focus as a professor, don't listen to anything they tell you in grad school, because they only know one way of how to be a professor. That is advice I got when I was finishing my bachelor's, and it was very much true.
Don't listen to people in academia. I have a question from your research actually. So you found the genes and you found these interesting phenotypes from the Vanderbilt University. Um, so do you have like a theory, a hypothesis of how that gene is affecting those specific phenotypes? Or you're like, I'm just leaving these to molecular biologists who want to do experiments?
I mean, mostly, yeah. I don't have enough. Well, I mean, we know that somehow through expression levels, they're affecting the phenotype. So one of the genes that I focus on is INO80E. It is a gene that's involved in chromatin remodeling. And as you might guess, chromatin remodeling could be anything and everything. It's a hugely understudied gene, that, yeah, I just don't have the data for figuring out a way it works. Um, Same with SBN. It's a gene from the immune system. So this is the first time having any sort of brain-related finding. Um, so there's it's very hard to make a hypothesis right now. Besides to say that, like, yeah, like if you're like chromatin remodeling is messed up, like yeah, of course things are going to get messed up. But that's about as much as I can say. There's also a lab that works on similar question in Drosophila, and they've also gone gene by gene to try to figure out what what causes phenotypes in Drosophila. But my gene happens to be the gene that they did not find a primer for, so it is unanswered
So
question.
they just skipped that experiment because they didn't have the primer for that. What about that initiative? I haven't followed that for a while, but Matthew State at UCSF was working with folks here at UC Berkeley in Drosophila, and they were like deleting the genes to see what would happen in development. Do you remember something like
I
this?
do remember that, but I can't say anything about it.
Yeah, because
Yeah.
I remember he was studying autism genes, and he was working with people at UC Berkeley to delete the genes and see what happened during development of the zebrafish brain.
Yeah, no, I think there's people at UCSF that are also just trying to catalog, as many developmental genes as possible. And I think, yeah, some of that work should be available to browse already. Yeah.
This may be a stupid question, but I don't know if you ever come across these people, but if someone tells you vaccination causes autism, what do you usually tell them?
Thanks for asking. No, it actually has not yet happened to me. Believe it or not. Um, I mean, the simple answer is that, the work that originally came to that conclusion was found to be, I think, fabricated, or at least had a lot of mistakes in it and then got pulled. And recently there's been a lot of initiatives. So Kaiser leads one of them, I know, that do these comparisons in huge groups of people, vaccination and autism, and they're confirming that there is no link of that sort.
That
Whether or not
initial
someone will find it.
study, that initial study was, I think his name was Wakefield, and they found out that he was being backed up by a group of attorneys in the UK because they wanted to sue the pharmaceuticals for the vaccine.
That's right.
Yeah, so you can find that information online. So they found that the this group of attorneys paid him for this study. He published it in a very well respected journal, which is Lancet. But then they figure out it was wrong because no one else was able to get to the same conclusions. So they retracted the paper. He is no longer a doctor since 2006, I want to say. I don't remember the year, but it's been a while that he's not allowed to practice anymore. The problem is when he published the paper, he was invited to give interviews in a lot of TV shows, and all those videos are still online.
Yeah. The loudest voices win. Yeah.
Yeah.
Um, but yeah, and I mean, I also think what one of the um, points I'll make that's not exactly related to that is that I think there's a very big gap between scientists and the general public. I think a lot of scientists are very bad at marketing their results and why they're doing it and what they actually mean. So I think there's a big need for this people that will go and bridge the gap between scientists and the general public, because Yeah, trust in science kind of depends on how things are communicated to the general public, for example, um, as well as um, willingness to participate in experiments. Like one issue that's also kind of tangential, right, is a lot of work we do in genetics is done, on basically white Europeans, white Americans, and it's very hard, to recruit other groups, like African Americans, Native Americans, because too much they've been wronged by the results of the science and how the science has been done. So there's additional connections that is important to make between scientists and various other communities.
How has this, like, I'm trying to formulate a link between that and your study. Like, have you looked into ethnicity in your groups, your controls cases, or is there a difference in mental disorders in different um, ethnical backgrounds?
Yeah. So yeah, I did not give that disclaimer in my talk. Most of the data that I present is on the white European population, um, with the exception of schizophrenia, where I also have East Asian population from Japan and Hong Kong, I think. And I can tell you that between the European Asian population, there was no difference in my study, but but a lot of these studies, yeah, we just remove all the people who are not white Europeans because there's just not enough people from other groups to make a statement about, but also enough people that we don't want that sort of confounding variable. Um, I'm not aware of population differences among brain-related traits. Those kinds of things are very hard to measure because they're very, cultural. Um, like even I mean, in many ways, like if you take like American millennials and Gen Z always talking about how they have depression, and you do not see that in older Americans and people from Russia or China. So it's very hard to measure.
So do they don't have depression or are they in denial? What's your hypothesis?
Because they're scared. Uh,
Yeah. So these behaviors are not only difficult to measure in mice, sometimes in humans too, right? If they're
yeah.
Chinese or Russian.
Uh, right. So it's but those are kind of like philosophical topics that um again, like those are kind of specific, I mean, that don't even take a small amount to solve. Those will take hundreds, thousands of people to help solve. I know.
Someone said it's definitely denial.
Are we doing pharmacogenetics in this class, or is that the other class?
No, it's the other class.
Okay, yeah.
Yeah,
Because
but
we would have more time to talk about those things.
yeah, I have a new topic of a project for you.
Oh,
You can compare a GWAS of those who voted for Trump and those who voted for Biden.
yeah, I mean, 23andMe might have that data. The problem is like 23andMe charges a lot for their data, so like a
You're
portal
cutting the low income, yeah.
does not have that. Um, but yeah, I need to go and watch those videos. So one of the kind of most controversial topics in genetics in the last year or two were studies on the genetics of sexual orientation. That this was done all from UK Biobank and 23andMe that had that information, and so those studies were done, and people are discussing, should those kinds of studies, be done, and what does that mean, for the results. And so last week I was at a conference. I haven't rewatched it, but we're going back to debate, that those kinds of studies in terms of like what sort of genetics should we kind of avoid studying, at least in this point in time. But I know, I think genetics of, people who vote for Trump, I think it's interesting.
Why is that a problem? I feel like if we can say, it's genetics, it's completely natural, like to have homosexuality. Does that make sense? For me, like homosexuality sounds natural because it's been like popping up everywhere in every kind of species. So what's the reasoning behind why we shouldn't study sexual orientation genes?
Well, because if we answer this question really well, right, what's that going to lead to? This is like in the very broad scheme of things, right? Genetic test for sexual orientation, that will that can either be done in utero, that that people might choose abortion for people just because of sexual orientation. Not, but I think the issue is not just in America, but in these countries where there's much heavier discrimination,
Like Russia.
against gay people, where if you're born and your genetics say, oh, like you're probably gay, well, that's gonna haunt you from birth. Um, but I mean, your argument is exactly right. It's exactly why we what the authors were hoping to gain from that study is to say, like, this is genetic, this is inherent, like, but there might be kind of additional implications. For example, here's another one that was kind of controversial. So they compare genetics of sexual orientation to the genetics of a bunch of other traits to see what the overlap is. And one of the traits was risk-taking behavior. And so that kind of feeds into a major negative stereotype that was coming up during the HIV crisis, that gay people were just kind of more likely to engage in risky behaviors, which I am not sure. I don't think there's any other sociological data to support that. But now, with that kind of genetic data, well, like, those stereotypes are also going to be coming back up. So those are kind of the questions that are raised by that study.
But then if you go back to why they are taking the risk behavior, there is something. I feel like there's probably like depression related to that, right? Where
Would you
you're
say?
having a disregard for your own life. Right, there's like something that's primary to that, and you're
Sure.
just looking at the consequence, and you're saying that being homosexual makes you take bigger risk, but you're not taking into consideration that being a homosexual in this society is going to make you more depressed. When you're depressed, you don't take good care of yourself as you should, and you're going to have more high-risk behavior because of that.
Yeah, that is something that we probably don't have data to disentangle. Yeah, especially these sorts of cultural things that very few of them that are going to be coded in genetics.
Okay, any more questions? I got excited. We got excited and went off topic.
No, I mean, I think, I'm always of the philosophy that those kinds of philosophical discussions are what you're going to remember more leaving this class than, the technical content. So I think that's totally cool.
Okay.
Um,
No more questions. And thank you for coming and sharing your work with us. Round of applause. We're all muted, but we're applauding.
Yeah, I mean, Erica can help you get in touch with me, if you need, and if you want to read the study on bioarchive, I could pass the link on. If you want to practice reading the scientific literature, which is also a very good skill.
Well, okay, thank you. Have a good rest of Thursday afternoon.